Hi, this is Dr. B. Thank you all for joining in. I was asked to do this webinar, and I was kind of at first excited, but then also scared because, as I learned through the years, relapsing fever is a very confusing topic, and the more I looked into it, actually, I understood the confusion, but I didn't get the answers that I wanted, at least some direction. And kind of one of the things we wanted to do, so I can get the slide to work. All right, just what we call a poll question. Which one of these 5 is actually a relapsing fever Borrelia? Ah, there it is. The answer is B. Turricaceae is the only relapsing fever Borrelia on the list. Now, the question was designed to kind of set the stage for this because, let me go through the next one. What actually is tick-borne relapsing fever? What is it? Well, over the years, there are different ways to define it. Sometimes it's been defined by a clinical presentation of these relapsing fevers, but then some of them don't cause relapsing fevers. It's been defined by the tick vector, the soft tick, but believe it or not, another surprise, some relapsing fevers are transmitted by hard ticks. It's been defined by genetics, and I think a lot of you have seen these genetic trees where it shows different branches and they group together different Borrelia. But, you know, you can do these branching trees based on different genes, and whether it's, you know, a flagellin gene or even a telomere gene, you're going to find different branches and different interpretations, so that's not so useful. It's also been defined by serotype, in other words, by surface antibody. You know, years ago, that's all the tools that the scientists had. They didn't have PCR and sequencing, so they did by serotype. And they would come up with a list, but then they found that it didn't really work because the serotypes changed. And that's a whole hallmark of tick-borne relapsing fever, is that it changes surface antigens on a regular basis. So they kind of gave up, and they defined it, or they actually called it and named it tick-borne relapsing fevers with an S, because they just said it's a group of these things, and we can't tell you what it is. And so that has made it kind of interesting and kind of difficult. But on the other hand, modern technology has helped us to define better what's going on, but not always. We'll go through it. So classic tick-borne relapsing fever, things you might have read about in a medical journal or textbook, say, a decade or two ago. It's an illness with recurring febrile episodes last about 3 days, then separated by afebrile periods of about 7 days duration. So it's a fever, then it goes away, and then it relapses and comes back and forth. Now, each febrile episode involves a crisis, what they call a crisis. There's a chill phase with high fever. In this case, 106 or more. I haven't seen it that high. I've seen it 104.5. During the fever, the people can become delirious and agitated, tachycardic, tachypneic, really, really, really immensely sick, and it will last up to half an hour. Then it changes to a phase where the fever breaks with drenching sweats and a rapid decrease in body temperature. And during this time, the patients can become transiently hypotensive. In fact, some people have gone through cardiac arrest during this phase because it's such a tremendous crisis. Classically, the teaching has been that even if you don't treat these people, they'll go through several of these phases, and then over time it just seems to go away. Now we know that's not always the case. But anyway, that's the classic thing. Now the cycles, what are they? As I mentioned before, the Borrelia express surface antigens, but in the classic relapsing fevers, the antigens undergo cyclic changes. And the cycles come about in coinciding with the regular variation of these antigens. And they have different names, VSP and VLP, a very small protein, very large protein. It's interesting. What happens is, because there's a switch of antigens on the surface, which is what the host looks at for an immune response, just as the immune response starts to form against these surface antigens, they switch. So the immune response is not killing these anymore. And so it goes through a whole other cycle, and the immune system changes again. So in the meantime, the organism has been changed, and it is evading this host immune response. Interestingly also, with every recurrent cycle and new surface antigens, as you would expect, there's a whole other group of late-appearing IgM antibodies. Now, as an aside, you know, we've all seen late-appearing IgM in Lyme Borrelia, but Lyme, you know, it may not be a relapsing fever Borrelia, but it does have 4-week cycles, and it does have changes of surface antibodies, antigens, I should say, to some degree, and we have seen late IgM, so that's kind of the basis of it. And in my lectures on Lyme disease, I say, you know, it is, you know, related to the relapsing fever Borrelia, so these late IgMs are not surprising. Because the genes that control the VSP and VLPs themselves vary, and there's a lot of polymorphism. It's helped these to remain in the community and not be overcome by host immune response, as what they call herd immunity. Makes a very interesting disease. All right, so we talked about the presentation, the classic presentation, but as I said, there are a lot of exceptions. In fact, one of the real big news deals about this whole thing, which is one of the reasons why I wanted to talk about this, is that relapsing fevers can actually present like Lyme disease. Now, there's a study of 543 United States patients who were tested thinking that they had Lyme disease, suspected Lyme. They were not having cycles of high and low fevers and relapses and all. They had Lyme symptoms, and guess what? Countrywide, 29% of these actually had tick-borne relapsing fever. And if you do a California cohort, which is, West Coast where these seem to be a little bit more prevalent, fully 38% of them did not have Lyme. They had tick-borne relapsing fever. So this is a very strong example that tick-borne relapsing fever can present like Lyme disease. Now, as an aside, you see in parentheses, when they tested these for tick-borne relapsing fever, they only tested for 2 species. And as I'll show you in a minute, there are a lot of more species here in America that we have to be worried about. So even with this narrow looking for relapsing fevers, in fact, more than a third of them really were relapsing fever presented like Lyme disease. Now, the other thing about it, so what if you have a patient who has what you suspect to have Lyme disease, you run a routine Lyme disease test and comes out negative, or maybe one band? Well, that's so-called seronegative Lyme, but maybe it isn't. Maybe it's relapsing fever. All right, let's talk now. The second thing that helps to define relapsing fevers, the vector, the soft tick. What is a soft tick? They're called Ornithodoros. These ticks are very unique. They have a lifespan of 10 to 20 years. They can endure starvation without feeding for as long as 5 or more years. And once they're infected, they will stay infected for the rest of their lives. At least one of them, I have this as a typo. It's not B. turricate, it's O. turricate. It's maintained transovarially. In other words, I'm sorry, it is correct. This Borrelia can go from parent through egg to offspring, meaning that the larvae themselves may be infectious and can transmit the infection. So this is one more reason why we have a problem. A little bit more about the ticks. The ticks are rarely seen because they don't attach and stay on like the Lyme disease ticks. They stay attached for as short as 5 to 30 minutes. They attach, they bite, they draw blood, and then they detach and they crawl back to their home. The bites, as you can expect, are painless, and this is a killer. Transmission of at least one of these, and probably many of them, occurs within seconds of the tick bite. They did a study with mice where they serially attached these ticks to the mice ears, and then they biopsied the ear every 15 seconds. Within 15 seconds, they were positive. So this is not something like Lyme disease where you have to think, attachment time of 5 hours, 12 hours, 24 hours, or whatever. And now, another corollary with Lyme, or another point. If you have a patient that says, I was out in the woods for a short time, and I came home, and I didn't have any ticks on me or anything like that, but for the next few days I got sick, and then I got a rash, and I think I have Lyme disease. Well, maybe such a brief attachment time makes you start to think maybe this is more relapsing fever than it is Lyme disease. Now, these ticks can bite a person again and again and again. They can feed multiple times. So that's why they live so long. They can keep feeding. Also, they don't live in the grass. They live in crevices and cracks. They can be wood cracks. They can be foundation cracks, leaf litter, caves in houses, and even cabins. They can live indoors, especially if the place has an infestation with rodents and other small mammals, because they live within the nesting of these animals. And once they bite you, 5 minutes or so, they crawl back in. Another thing I learned is that, like many other ticks, they're attracted to heat and carbon dioxide. And very often, people with cabins or go camping, they turn on a campfire or a wood stove, turn on the heat in the cabin, and out these things come. They bite people when they're resting or asleep. So it's a pretty unique and nasty tick. I'm showing you pictures here, and you can say, well, this is so different from a Lyme tick. It should be pretty easy for someone to see this. That is assuming you can, because it's only 5 minutes it's on there, and usually when you're sleeping. But look at the next slide. This is an engorged Ixodes tick. Now, I would have problems differentiating an Ixodes that's engorged. Let me go back to one of these. Yes, there are differences, but, I don't know, not all ticks look alike, and these certainly can be confusing. So again, going all the different ways, you think about, okay, this is relapsing fever, not Lyme. It's a different tick. Well, maybe it is, maybe it isn't. So these ticks are not unique. For example, miyamotoi, we know that it can be transmitted by ixodes, so that's not a soft tick. Lone Star, the amblyoma, is not a soft tick that it transmits. I've read some recent articles I haven't put on the slide where there are different relapsing fever Borrelia found in Europe that are definitely related to hard ticks. So again, defining relapsing fever by the kind of tick is not necessarily ironclad. No surprise that they have complex genetics. Sequencing is done specifically on a specific gene, for example, flagellin gene and other genes, even telomeres. But as I said earlier, the different family trees that result make these line up differently. So that kind of thing is interesting for researchers, but it does not help us as clinicians. Now, serotyping, as I mentioned before, is based on the expressed surface antigens, but that's tricky because of the antigenic variation. So to be really accurate, you need to measure multiple types of each surface antigen. In fact, that's the basis of advanced immunoblotting, and also the reason why standard non-immunoblot serologies are usually very inaccurate. Here's some basic facts that are important for clinicians. As I said, transmission within 15 seconds of a tick bite with at least some of these guys. In terms of maternal fetal passage, it's always controversial in Lyme disease for whatever stupid reason. But here it's well recognized and accepted. Spontaneous abortion, premature birth, neonatal death are all recognized consequences of maternal infection. Now, the louse-borne relapsing fever is different from tick-borne, and in this way, these Borrelia are transmitted by mucous membranes. In other words, people find a louse and then they scratch it off, they squash it or whatever, and if their finger touches their eye or their mouth or another mucous membrane, they can get infected that way. Now, we all know that ticks, when they're on you, they are not clean. They leave behind feces, which, excuse me, in some cases are infected. So I can understand that this might be a reason for transmission of a relapsing fever in someone. So not proven in the literature, but something makes my antennas go up. Another important point, ARDS, acute respiratory distress syndrome, has been associated with Borrelia hermsii, which is relapsing fever Borrelia. This is according to CDC. Number of case reports of that. Another really interesting thing, the Synithodoros tick. It lives so long. It has, some of them are born infected, can live for 20 years, 10 years. It serves as its own reservoir. In other words, it has a Borrelia in it. It doesn't need to feed on a mouse or a deer or some other thing to maintain or get the infection. It's its own reservoir. It doesn't have to have a mammal as part of the life cycle. That's really unique. Another thing about these species, some of these Borrelia are immune to complement-mediated killing. One of the articles I read showed this, we know this for Lyme disease, but they actually measured the effect of this, and it's more pronounced in some of the relapsing fevers than even in Lyme Borrelia, meaning that that could be a reason for the more acute infection and why, and I'll get to this, relapsing fevers can become a chronic infection the way Lyme Borrelia can be. Another important point, prolonged QT interval has been reported with these infections. Maybe that's related to the cardiac arrests that have been seen in the acute cases. So do we need to worry about this? I mean, I'm telling you scary stuff being a medical term. So do we need to worry about it? Well, yeah. Here's some quotes from one of the articles. They said, TBRF is typically considered a disease of outdoor enthusiasts and impoverished, nations, primitive conditions. But they did a study that suggests that one of the Borrelia, toricate, lives in metropolitan areas. In fact, they call it endemic. They found it in San Antonio, Dallas, and Austin, the 7th, 9th, and 11th largest cities in the United States are endemic for this kind of a tick. Another point, 58% of turkeys that were collected during the fall hunting season about a decade ago were tested positive for B. miyamotoi. So it's really out there in the environment. It's not some obscure thing you find reading about caves of Africa or something. It's really where we are. This is a pilot study that was printed or published a couple of years ago, and they found case reports of miyamotoi, hermsii, lone star, parkeri, toricate in the United States. Another species, coriaceae, is found in the ticks. Have not reported it yet, as far as I can see, in in humans, but I would not be surprised if some show up. Well, the reason for this slide, showing the high prevalence of the co-infections, is to make a point that, again, relapsing fever infections are not that rare. This is a review of 10,000 patients who were tested for tick-borne disease. If you go to the box on the left, percentage-wise, 37% showed Babesia positive in one of the tests. They did all different types of tests, serologies, PCR, and so forth. Burgdorferi, 32%. Tick-borne relapsing fever, not that much lower, 27.7%. So again, it's almost as prevalent as Lyme disease if you start to look for it. Again, Lyme serologies, classic old-fashioned Lyme serologies, will not necessarily pick up tick-borne relapsing fever, or at least not with a strong enough reaction to be called positive. So you might be missing a lot of people. And maybe this accounts for the so-called seronegative Lyme case where we see our patients, maybe 2/3 of them have a positive Lyme serology and 1/3 of them don't. Well, maybe those are the relapsing fever patients. Box on the right just emphasizes that 2 infections, 40%, 3 infections, 15%, and so forth. I mean, it's not rare, as you all know, to have co-infections, and you can have a co-infection of Lyme Borrelia and relapsing fever Borrelia. Studies have shown that. And here's that slide. 543 patients, this is from that earlier study. 32% had Lyme antibodies. 22% had antibodies to tick-borne relapsing fever, even though they only tested for 2 of the species. And 7% had both. In the California cohort, the numbers are similar, but a little bit worse. A little bit higher percentage of antibodies to relapsing fever Borrelia, and 11% had both. That's been reported in 49 states. In some states, like Lyme, it's not a reportable disease, so these statistics are probably conservative. But, you know, there it is, Alaska, Hawaii, all across the United States. I see Utah as the one state that doesn't have a lit-up county, but knowing the terrain of Utah, I wouldn't be surprised if it's there as well. And it's been found in Central and South America, Europe and Asia, Africa. There's controversy about it being in Australia, but there are reports of it being serologically detected there. And, you know, people in Australia don't necessarily like to hear that, but that's the fact. Now, clinical presentations. As I mentioned, there are 2 types, the classic presentation and the Lyme-like presentation. The classic one is acute, fever, chills, headache, myalgias, arthralgias, nausea, but hitting really hard, really fast, and lasting for a while, and then seemingly getting better. Now, this can really confuse an emergency room doctor. Someone comes in, oh, it's a viral illness, you'll be better in a few days, and sure enough you are. Then a week later they come in sick again, they say, oh, and you've got another virus. Well, you know, it may not be that virus, it may in fact be this. But the Lyme-like presentations, as we have seen and maybe have experienced with our patients, it includes a whole gamut of early Lyme. Some actually do cause rashes. Intermediate, long-standing, even chronic cases. I know many, many years ago there's a case of relapsing fever. A patient had very severe multi-system disease and ended up with endocarditis as well as epicarditis. They had a cardiac effusion, a pericardial effusion, and they were able to culture relapsing fever from that effusion. That patient had chronic arthritis, encephalitis, and peripheral neuropathy as well. Now, miyamotoi is interesting because it seems to be a blend of the 2. As I mentioned before, it can be found, or is found, in hard ticks, so that's a little bit different. It has symptoms, I'll show you in a second, that include from both categories, and that's why you might have seen it being referred to as Borrelia miyamotoi disease. They're trying to give it a separate classification. I think, my personal idea is I think it's a little bit too fuzzy to make it a whole separate disease. Start calling, you know, Borrelia miyamotoi disease, have to start making new diseases with every species, and since they blend, you're not going to have a good time of doing it. Trying to reinvent the wheel of saying relapsing fevers. But anyway, that's what this refers to, that there's a blend of the 2. One other subtlety, if you look at the statistics, it's more likely that patients will complain of GI involvement, abdominal pain, nausea, vomiting, than they do with Lyme Borrelia. But because of the acute presentation of tick-borne relapsing fever, you know, not as only an acute virus, but you might confuse it with rickettsia, babesia, malaria, which has happened in Africa, and even tick encephalitis viruses. So you just have to have your antennas up and look for good testing, which is another part of the speech today. This is a compilation of some of the symptoms from different studies, CDC as well. And for miyamotoi, you see fever, fatigue, headache is highlighted as really being present in a lot of them. When you go across along the side, relapsing fever, Borrelia, fever is very common. As you go to Lyme, it gets less common, Lyme, Borrelia, because as you know, not every Lyme patient gets a fever, and sometimes those who do, we start to think co-infection right away. But fatigue is common in all of them. Headache, yeah, but look down. Chills, miyamotoi less so and more like Lyme than relapsing fever, and that's where we start to get this overlap of the two. And you go down the list and you can see what I'm talking about. Also, we can go down to vomiting, second to the bottom. Relapsing fever, Borrelia are very commonly having this, vomiting and nausea, whereas you don't see that in miyamotoi and not in Borrelia for Lyme. So that's why this miyamotoi disease was brought up, but just so that you know there is a wide spectrum of this. Now, so far, and I say so far, we can identify at least 18 different identified species of relapsing fever Borrelia, and I keep emphasizing so far because it seems like every year they're developing or discovering new ones or renaming old ones and splitting old ones into 2 or 3 categories. So just to emphasize that there's a lot of them out there. And if you gave them each their own name, Hermsii disease, Lone Storii disease, miyamotoi disease, it didn't start to make you dizzy. So right now, just this is FYI. Luckily, in the United States, we don't have all of those that we can say are big clinical problems. What we've seen so far based on advanced testing are the 7 here. Number 7 has a question mark because it's not really sure whether this is its separate species or one of the others, a subspecies, a strain of one of the others, and that's being debated right now. Now, how do you test for this? Classic tick-borne relapsing fever, classic testing is by doing a blood smear. And indeed, if you can get the blood during an acute febrile phase, you can in some cases see the actual spirochetes. This is a real laboratory slide showing the Borrelia on the blood smear. In tick-borne disease areas, blood smears are often done when people present with a high fever because they look for Babesia parasites, and they often find this, when they find it, as kind of a surprise, like, oh, we didn't look for that, we weren't looking for that, and this is what it really is. It's not Babesia after all. So you can see it on blood smears, but it's only during the acute stage of the crisis, during the chilling and fever going up phase. Obviously, you can't determine species from this. And, in my work with Alan McDonald and Eva Sapi doing Borrelia cultures, we found a lot of squiggly things in the blood that looked like this and moved like they do under the microscope, and turned out not to be Borrelia. There were cytoplasmic strands from usually from the white cells. There were also little strands of collagen. So just looking at this to make a firm diagnosis is not always helpful because it could be a false positive. Now, also we've heard of people having labs that say, I can do a dark field smear and I can see spirochetes and I can make a Lyme diagnosis right away. And if you can't see it, it's because you're not looking right or you don't have a good microscope. I mean, you hear this over and over again. Well, maybe they're not seeing Lyme. Maybe they're seeing relapsing fevers because the concentration of Borrelia in the blood during the acute phase of relapsing fever Borrelia is hundreds of times higher, maybe even thousands of times higher than what happens in Lyme Borrelia. So blood smear had been the old standard because that's all they had. Nowadays, we can get PCRs. Large commercial labs, you can get a PCR for miyamotoi only, and that's it. You can't for any of the other ones that I mentioned, the 6 or 7 others that we have in America. IGeneX does offer a PCR. Theirs is interesting. It's genus specific. So they might give you a positive and say, you have a relapsing fever Borrelia. We can't tell exactly which one it is, but we know that one came out positive on the PCR. They are able to definitively say miyamotoi based on the work that they have done. But nevertheless, it's much broader than the standard commercial PCR from, say, a LabCorp or a Quest. Trouble about PCRs, just like in Lyme disease, the sensitivity is low. In this illness, the sensitivity is good really only during the acute stages of the disease, like when you'd see them in the blood on the smear. Also, in those who are immunosuppressed for whatever reason, and those who are very ill, late-stage patients whose immune system often is suppressed or poorly functioning. So PCR has a definite role, but it's not the most sensitive test out there. Serologies, they can be problematic. A lot of TBRF Borrelia express P41 that can give rise to false positive Lyme ELISA, and sometimes we'll see what we think is a Lyme patient, and on the Western blots it's just band 41. So, well, it probably is a false negative or a weakly positive. The patient seems to have Lyme, and I'm going to go with the 41, knowing that 41 is very nonspecific. Well, maybe in this case it really was relapsing fever, and you didn't know it. an unexpected TBRF infection. Interestingly, OSP-C, or something that migrates along with OSP-C, is present in several TBRF species and can give you another potential reason for a false positive Lyme serology in some labs. Doing serology for relapsing fever in a commercial lab, again, one of the ones that most people have, Quest, whatever, they'll do Hermsii and nothing else. You can get what's called a GLP protein-based ELISA for miyamotoi. GLP is only one protein antigen that's associated with these relapsing fever Borrelia, so because not all of them express it, and if they do, they might express it weakly or have different serotypes, you can get a lot of false negatives if this is the only thing you rely on. It's just like doing a Western blot looking for one band, some oddball band that hardly ever shows up. It's not the greatest way to do it. IGeneX now has a lot of new things based on serology. They have an Immunoblot, which I've talked about before, and I'll give you more details in a moment. It gives you really the most information. It's, at this point, definitely the gold standard, and a lot of good reasons for that, and I'll show actually good pictures. They have a simpler test, a Broad Coverage Antibody Assay, that's kind of like a simple and cost-effective like an ELISA would be, but still much better than what the large labs offer. And I'll show you pictures of that one, too. There you go to the Immunoblot. Here we go. What is it? It uses recombinant antigens. These are created in the lab that are specific to multiple individual species. So we're not looking for just one or two, we're looking for multiple species. In a Western blot and in other types of blotting, the antigens which are not recombinant are spread out on a gel and they migrate with electrophoresis and they end up where they end up. Here, and where they end up is not always precise, and how much is there is not always precise. But in Immunoblot, it is, because they deliver exact amounts at specific locations, and they know exactly what's going on. Again, I'll show you pictures. And in the Immunoblot, instead of using one protein antigen like they do for the GLP test in the commercial lab, they give a large variety of protein antigens, not just one. And they do use it for the 6, actually it's 7, but it's usually 6 most commonly found species, not just Hermsii or miyamotoi. Now, what about the recombinant antigens and all this? It makes it much more sensitive because you're picking up all the different species that are important, and much more specific because these are recombinant antigens and not things that just migrate based on molecular weight as they do in Western blots. So they're less likely to cross-react with viruses, non-Borrelia bacteria, and other autoantigens like an autoimmune disease. And what's also very important is that these will not cross-react with Lyme testing. So if you have a patient with Lyme and you do a TBRF Immunoblot, they're not going to show positive. There's no crossover. Also very important, as I mentioned, relapsing fevers give IgM responses even late into the disease. That's recognized. So importantly, these Immunoblots report IgM and IgG, very important. So again, Western blot, how to identify the bands? Well, the position is based on migration, and the migration is not exacting. The variable location of the bands can make it very hard to line up and identify what's really going on. And because we're using electrophoresis here, unimportant and non-specific proteins may co-migrate with the Borrelia ones, and the Western blot can't tell you which is the real one, which is the not real one. And the scoring of this is a positive or negative based on how dark, how intense, how black the band is. But who sets the standard? And how dark is positive? How indeterminate is indeterminate? Protein content varies from test to test. The band's intensity can vary. Hard to read these. Also, if you get a broad band, like look at this, the picture on the right, there's a part like 2/3 of the way up and there's one toward the bottom, really broad band. What is that? Is there more antigen in this person's blood? Is there a lot of co-migrating antigens that seem to stop at that one spot? Is there a stronger host reaction? Who knows? Look at this. This is an actual Immunoblot, this picture. Look at these precisely drawn bands in exact locations to know exactly what everything is, and they're put in specific spots. They're not spread out based on some migration. They're put in specific spots, this exact amount that they measure and put there with their machinery. So you know exactly what these refer to. And see, for example, GLP, which is what they did in miyamotoi in the commercial lab, I see 1, 2, 3, 4 different ones that they're looking at because of the strain variation and species variation of these GLPs. And if you don't do that with this and the other ones, you're not going to pick up what you need to pick up. So because of all this, you're not going to get the false readings with viruses, other bacteria, autoantigens, and so forth, giving you less false positives, less false negatives. And just put them side by side to show you this. And the Immunoblot on the right, that's not a drawing. That's the real Immunoblot. That really is what it looks like. So huge difference, huge difference. And if I was a lab person trying to sit and identify these things and look at the Western blots and come up with some kind of an answer that's important for the health of somebody, I would feel nervous. But with an Immunoblot, I think it's much more secure. I'd rather be an Immunoblot reader than a Western blot reader. So what they offer here is a set of 6. That's the latest iteration of it. And because it's IgM and IgG, those 6 species relate to actually 12 different individual tests come with one Immunoblot for TBRF. So you're really getting a broad coverage of what's out there. And this is an example of what's being seen. It took people who were positive on the Immunoblot, 7 were Hermsii, 62 of them, 7 are Hermsii, 13 miyamotoi, 13 toricate, 2 Turcica-like, and we say Turcica-like because it's not exactly well worked out the genetics of this, but it looks like Turcica. Very bottom, Hermsii and miyamotoi together. What about the one up from the bottom, TBRF species? They found 26 species. What does that mean? Well, these things, as I mentioned, they have variable antigens. There are a lot of different species out there that overlap. And rather than having a negative test, this will actually report that there's some species out there. We can't exactly identify which species, but we know it's a relapsing fever Borrelia, and there it is. So I don't think showing a species and not speciating it here is a disadvantage. I think it's a major advantage because it shows you that there is something there rather than give you to say it's negative, as another lab might do. What about sensitivity? In early disease, when you don't expect things to be positive, they nevertheless found a sensitivity of 66.7%. In those same patients, convalescent sample, 100% sensitivity. They picked up every single one of them. And these are patients who were confirmed to really have this illness by PCR. So this is a real result, 100% sensitivity of these PCR-positive patients in convalescent sample. Specificity, shows all the different things they look for with these test sera. To go down to the bottom, specificity is 98.6% for IgM, 98.6% for IgG. Yeah, sounds like a temperature. And when you combine the two, 97.6. And it's interesting that some of these are like syphilis and multiple sclerosis. You would expect those kind of false readings, but you would definitely not confuse the two with and CMV, allergy, so forth. You're not really confused with relapsing fever. But multiple sclerosis, who knows? Maybe that's Borrelia related in some patients. That's one of the controversies. Well, we can give a summary. Most important thing to realize is that it gives us information that really has not been previously available. You could not get a serology for some of these other Borrelia that's really out there. You just couldn't do it. If you look for Hermsii and that's all you look for, you get false negatives when it might be Coriaceae or one of the other ones. So it gives you information you just can't get anywhere else. Because it doesn't cross-react with Lyme, as I mentioned before, you really can narrow down what's going on. Because you can identify a lot of different species, it helps you clinically. You might start to find over time that, this type of relapsing fever Borrelia is different from that type, and I'm getting a better clinical feel for it. What we don't know, we'll go through treatment in a minute, but we don't know if one species responds to one group of antibiotics better than another. So by your doing Immunoblots and keeping the records, you're going to start to develop this information and become better doctors for it. As I mentioned, it's useful in early disease. I mean, if you have some of these ups and downs, fever, and you don't know it's an acute virus, and you might see something on a blood smear, it's nice to have a confirmatory serology that has such high specificity. And because it's a serology, you don't have to wait 2 or 3 weeks to get the results. It comes over fast. They have another test, which, as I mentioned, is similar but simple. It's called a Broad Coverage Antibody Assay. It's simple, cost-effective. Think of it as a better ELISA. It definitely replaces standard serologies because it does cover the multiple clinically relevant species, does include IgM and IgG as a single result. So you get a yes or no. So you don't have to sit and worry about bands and count bands and look at pictures. You just got a report that says yes or no. And again, being a serologic test, it's a rapid turnover. And when they say cost-effective, we're talking like probably in the range of what an ELISA would cost. So people who have insurance issues and financial issues, they can't spend the money on Immunoblot. This is a cost-effective alternative in some cases. I still think the Immunoblot is what I would order if I could do it, but they're giving you an option. So when do you use these new things? The Broad Coverage Antibody Assay is a more sensitive, highly specific, and broad-spectrum alternative to the standard serologies because it does test for multiple relapsing fever Borrelia species, not much more expensive than the standard ELISAs that are out there, and very simple to interpret, and includes IgM and IgG. Now, the Immunoblots, on the other hand, they really are the gold standard. You know, they're broad multi-species coverage. They can identify species, not in New York State, unfortunately, yet, but that's in the works. And as I said before, you know, as a clinician, I really want to know, I mean, this is how over the years we figured out, hey, it's not just Lyme disease, there's a thing called Babesia in these patients. And hey, there's also a thing now we're looking at what's called Bartonella. And it's a Bartonella-like thing because there's not one Bartonella, there's several Bartonella. Over time, we're going to start to get that feel for this and say, wow, this type of relapsing fever acts a little bit differently than that type. And this seems to respond to treatment a little bit better than that type. And it needs a little bit different antibiotic. So over time, we're going to become better doctors because of this. Sensitivity is incredibly high, very specific. And in early cases, as I said, it'll pick up at least 2/3 of them. So there's really no reason to do the other serologies if you can, because this is the gold standard. So when do you do what? Because you can do serologies and you can do PCRs. In someone who has intact immunity, the Immunoblot is always the first choice. It's good for all the stages of the disease. It does have IgM, which is still significant even in late disease. They report band groups, and 2 or more is reactive, is read as positive if they're reactive. 1 group is considered a borderline result. And they found in their studies when they had these borderline patients, by waiting a few weeks and repeating it, they would get a more clear result. So if you have 1 reactive band group, you just repeat the test a little bit later on, and you might get a much more clear picture. Now, if someone's immunocompromised, in addition to doing the Immunoblot, you would want to add the PCR. Interestingly, there's a reciprocal relationship with PCR and immune-based testing, because if the immune system is intact, the antigen load, the spirochete load, is generally lower, but the antibody level is higher. On the other hand, if the immune system is weak, you're less likely to have reactive antibodies, or at least free antibodies, but you're having more germs, and therefore the PCR is more likely to be positive. So by doing both, number one, you have a much better chance of picking up the real disease, and on the other hand, gives you a good picture of the immune responsiveness. So if you're someone who's seronegative but PCR positive, then you start to think about, all right, I'm going to change gears here, and besides the infections, what's wrong with the immune system? Let's start doing an immune workup and see what we can do to boost it. So usually very important to add both if you can. Now we talk about doing test panels. I'll go back to the slide I showed you before. These are 10,000 patients referred for tick-borne diseases. Look what's there. More than a third had Babesia. About a third each had Borrelia, burgdorferi, and relapsing fever Borrelia. You put them together, it's 50-60%. Almost 20% had Bartonella, Anaplasma, 16.7%, Rickettsia. You know, we don't think much about, you know, good old Rocky Mountain spotted fever, but maybe we should. I had a patient years ago, never suspected, and showed up. And thank God, because it helped the patient get over it. So when you do the testing, do a test panel. Don't just look, say, it's Lyme disease, I'll do the Lyme test. Think of the co-infections. And again, from the bottom box, at least 40% had 2, 15% had 3, 4.6% had 4, and even 5 within 0.7. So it's real. Well, this is kind of repetitious. Nearly all Lyme patients, even the more ill ones, have co-infections, as we all know. The high prevalence of TBRF, which may present acutely and with high fever, make it even more important than ever to look for the Babesia and the Rickettsia, especially because those two can make you really sick, even be fatal. Another thing which I've seen in the literature, there are a lot of reports, and actually it's pretty well accepted, that TBRF does involve central nervous and peripheral nervous system. So not only is that another corollary to Lyme disease, making it harder to differentiate them clinically, also makes it difficult to separate the relapsing fevers from people who have tick-borne viral encephalitis. Especially if you don't make the diagnosis of TBRF, don't treat it. It does become chronic, and you can have chronic neurologic symptoms, and just like a chronic encephalitis, you might not know what you're really doing here. And as I mentioned before, positive blood smears for spirochetes or protozoa require more definitive lab testing for confirmation, because what you see on a smear as a spirochete may not be. Treatment. Here we go. You ready? Question mark. As far as we can tell, the treatments for the TBRF and Lyme are pretty much similar. Unfortunately, there are very few studies on antibiotic susceptibility, and those that are out there are 40 years old. I mean, really, you know, they talk about tetracycline, and that's it. But there are patients who have known TBRF who really needed the IV antibiotics, long-term oral antibiotics, combination therapy, and so forth. So the points are, it can be a chronic illness. It can and does involve the central nervous system. It can involve the joints, and it can induce fatigue that is chronic if it's not properly treated. Now, a very important word of warning. In the classical presentation of TBRF, if you treat them during a crisis, severe Herxheimer's are known to have happened, including such hypotension and calamities, including cardiac arrhythmias, that there have been fatalities. And what's been recommended is that these patients be put in an ICU and be monitored if you're going to give them aggressive treatment during a crisis. So that's just a heads up. Scary stuff. So we're moving along here. I got some concluding thoughts. It's newly recognized as being far more prevalent than previously thought, may be responsible for many more cases of what we call seronegative Lyme. It's much more clinically difficult to identify than I had been thinking, especially without this laboratory confirmation. I recommend Immunoblot really because this way you can learn patterns and help you figure out diagnoses, treatments, and so forth. And to do that, keep records, share your information, and publish it all. So, you know, even if it means a simple abstract at one of the meetings, but get your stuff out there. I know you like references. This is an article that was published just a few months ago that talks about the making of the Immunoblot and all the different species that go into it and why and how and whatever. And so this one article is not only a good article to have, but the references in this also serve as kind of a library for you to learn more as time goes on.